Hello, my name is Professor Hudson, and you are about to view narrated PowerPoint lectures for Biotechnology Chapter 14. Let us continue with our discussion of cloning, which is another common biotechnology technique. In the previous segment, we left off with talking about cloning of genes, and the example used was the insulin gene. Now let's talk about the cloning of a whole organism. Now we have the ability to clone whole organisms such as sheep, cats, dogs, and other animals. One of the first organisms that was cloned and was documented to have been cloned was the sheep dolly. Basically, in this example, instead of just cloning in an individual gene, we actually cloned a whole organism. In order to clone the whole organism, basically what they did was a donor sheep's mammary cell is extracted and grown in a tissue culture flask. So they took one of the sheep's body cells from the mammary glands or the mammary tissue, which is where the breast milk is made. Then they took another sheep's unfertilized egg and removed the nucleus. Remember, in an unfertilized egg, the nucleus is going to be haploid, in that you cannot start to make a baby from this egg unless it becomes diploid through the fertilization of sperm. In the case of cloning, however, they took the diploid nucleus from the mammary cell and then put it inside of the unfertilized egg that had had its nucleus removed. So basically they replaced the haploid nucleus in the unfertilized egg with a diploid nucleus from a body cell. So now you have an egg that has the full amount of chromosomes it needs to develop into a baby without actually having been fertilized with sperm. Now, once this unfertilized egg with this diploid nucleus has been constructed, they use certain chemicals and materials in order to cause this unfertilized egg with this diploid nucleus to start developing by mitosis into an embryo. And then after a small number of cell divisions, they took this embryo and put it into a surrogate sheep. The pregnancy was allowed to proceed, and then this sheep gave birth to a baby lamb called Dolly, who was actually a clone of the sheep for which we got the mammary cell and the diploid nucleus. So Dolly is a clone of the sheep that we first started with, for which we got the diploid nucleus from. Now let's talk about some specific biotechnology products that were created using recombinant DNA technology and genetic engineering. Also, sometimes cloning is used in these procedures. As we mentioned, transgenic organisms are organisms that consist of DNA from different species. Recombinant DNA technology is used to produce transgenic bacteria, transgenic plants, and transgenic farm animals. Transgenic organisms, often called GMOs, can be used to produce proteins or hormones such as insulin, antibodies, insect-resistant plants, drugs, whole organs, etc. In the previous segment, you saw how an insulin gene could be cloned into a bacteria in order to produce large amounts of insulin protein. In this slide, we're showing another way in which you can use biotechnology in order to produce insulin for people who don't make it themselves. In this example, we can use recombinant DNA technology, genetic engineering, along with cloning in order to insert the human insulin gene, or in this case the human growth hormone gene, into a donor egg and have it develop into a baby goat that can produce the growth hormone or the insulin in their milk. This is another example of genetic engineering and recombinant DNA technology being used to create a transgenic organism. The organism is considered transgenic because you have a goat with goat DNA that is now carrying genes for the human growth hormone, or human insulin. And so the goat has human DNA mixed in with its goat DNA. And so it's transgenic. The next few slides talk about some other common applications of biotechnology that you may run across. So genomics is the study of genomes of humans and other organisms. Understanding the genomes of humans and other organisms is critical if you're going to be doing genetic engineering and recombinant DNA technology, where you are manipulating the DNA of humans and other organisms. Bioremediation is where microorganisms are used to decrease pollutants in the environment. For example, if there's an oil spill and bacteria are engineered to dissolve the oil so that it can be cleaned up quicker, that would be an example of bioremediation. Molecular farming. Molecular farming is another example of biotechnology. This is where you can produce medically important proteins in the livestock of mammary glands. So the example of the goat producing growth hormone or insulin in its milk would be an example of molecular farming. On the next few slides, we'll talk about some other biotechnology tools and procedures that are used sometimes during the process of biotechnology. Oftentimes, in vitro fertilization is used in biotechnology. In vitro fertilization is when the eggs are removed from the woman's ovaries, the sperm are removed from the man's testicles or through his ejaculate, and the sperm and the egg are put together in a tube where fertilization occurs in a tube or a petri dish. This can be used during cloning procedures and also can be used with infertility when couples cannot get pregnant on their own. So in vitro fertilization is where the sperm fertilizes the egg outside of the female's body. Once the sperm and egg get together and fertilization has occurred, then the embryos are put back inside the woman, where hopefully they will attach to her endometrial lining and grow into a baby. This is another slide just showing once again in vitro fertilization. Typically the embryo, and it's not even called an embryo at this point, but typically the little ball of cells is put back in the woman about 24 to 48 hours after fertilization in the test tube. Intra-Cytoplasmic Sperm injection is another technique that can be used in biotechnology, in particular when creating different types of organisms. It's also used in couples who are having infertility issues. In this case, this is often used where if the infertility issue is with the male, like let's say the male is not producing sufficient sperm in his ejaculate, or he's producing sperm that for whatever reason cannot swim. The doctor can actually take a slice or section of his testicle, remove some of the sperm directly from the tissue, and inject it into the woman's egg. Then it'll be allowed to grow into a little ball of cells in a dish, and then about 24 to 48 hours afterwards, it'll be put back inside of the woman. Amniocentesis is a procedure where you can look to see what the chromosomes look like of the developing embryo or fetus, and this can be used sometimes in conjunction with genetic counseling, and there are other applications in biotechnology as well. But during amniocentesis, a long needle is used to withdraw amniotic fluid containing fetal cells, and then biochemical tests can be run on the amniotic fluid, and the chromosomes from these cells can be analyzed. And some of the disorders that you can detect with this procedure is you could tell if the child was going to have Down syndrome or some other chromosomal abnormality that results from non-disjunction. Chorionic villi sampling In this procedure, a suction tube is used to remove cells from the chorion where the placenta will develop. This chorion membrane has some of the fetal cells in it, and so you can analyze the chromosomes from this as well. This procedure has the advantage that it can be done at 8 weeks, but it's usually done more like at 12 weeks, whereas with the amnio, you typically have to wait until you're at least 16 weeks to do the amnio. The problem with this procedure, though, is that there's a higher rate of miscarriage and a higher rate of genetic mutation with this procedure, only because you're not just taking fluid, you're actually disrupting some of the tissue in the placenta attached to the developing baby. Gene therapy, a lot of times when we talk about biotechnology, also we talk about gene therapy. Gene therapy involves procedures to give patients healthy genes to make up for a faulty gene. It also includes the use of genes to treat genetic disorders and various human illnesses. There are two ways in which you could do gene therapy. Somatic cell gene therapy is where you treat the mutation within the person's body cells. This could potentially cure them of the disease. However, it does not prevent them from passing the gene on to their offspring. However, if you do germline gene therapy, this is where the mutation is corrected at the level of the sperm or the egg, or early in the development of the embryo. And then in that case, you not only can cure the problem in that person, but you can also prevent it from being passed on to future offspring. A lot of times there is discussion about the ethical and or bioethical issues in doing this type of work, where you are treating genetic mutations within the parents or early in the embryo, and some people have issues with that. And in the book they talk about some pros and cons of doing gene therapy and whether or not people should be allowed to basically design their babies in that they are correcting or changing things in the baby before it's born. The last thing I'll talk about is related to biotechnology is stem cells. Stem cell research that's something that is a hot area right now. Basically, all cells in the body come from stem cells. A stem cell is a cell that has not yet taken on a specialized function. Stem cells are special in that stem cells can self-renew to make more stem cells, and they can differentiate into specialized cell types. Some terms that you might hear are terms such as pluripotent. Stem cells that can become many types of cells in the body are called pluripotent. And then stem cells that can only become a few types of cells are called multipotent. One of the reasons there's such a lot of debate about stem cell research is that the type of stem cells that really hold the most promise are stem cells that are what we call totipotent. Totipotent stem cells are stem cells that are removed from a newly fertilized egg about 8 cells into development. So about 3 days after fertilization, the little ball of cells have cells that are what we call totipotent. Each one of these cells has the potential to become any cell type in the body. And these are the ones that most people would like to get. However, there are ethical issues about creating embryos just to get the cells at this stage. Pluripotent ones are also good, not quite as good, but these cells have the ability to become many cell types in the body as well. But these are not as useful as the totipotent cells. Because of these issues, most of the time when stem cells are obtained, they are obtained either from umbilical cord blood or from adult stem cell sources. And these types of stem cells are useful, but not as useful as the totipotent or pluripotent stem cells. This concludes our narrated PowerPoint lectures for Chapter 14.